Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Tamar Geffen, Associate Professor at the Mesulam Center for Cognitive Neurology and Alzheimer's Disease at the Northwestern University. Tamar is also a 2023 Toffler Scholar. Today, we will be discussing her focus on neuropsychological characteristics of dementia syndromes and other age-related disorders. So let's dive in. Welcome, Tamar.
Thank you.
I'm super excited to have this question because you are doing some very interesting things. And besides that, you're just a very interesting person. So I'm very grateful that you're making this time. So I would like to just start a little bit about your journey. Like, how did you get to where you're at, and what was some of the influences?
Okay. Well, first, thank you for having me. I'll start off by saying that I'm a clinical neuropsychologist. I'm someone that is really in love and passionate and enamored by the human brain. And so the journey for me, actually, like everyone else, it started up at home. I grew up in a religious and relatively observant home and community. Lots of laws that I didn't necessarily follow. A lot of scripture, analysis, and emphasis on the spiritual, on God. I was a skeptic even as a child, and I still am. I've learned to now accept it and be proud of it and turn it into scientific inquiry, I think. I'm still working on it. There was this quote that always resonated with me by Carl Sagan. It's a popular one, but he was describing his parents' influence, and he said, "My parents were not scientists. They knew almost nothing about science, but in introducing me simultaneously to skepticism and to wonder, they taught me the two uneasily cohabiting modes of thought that are central to the scientific method." And my parents were not scientists, but they really pushed inquiry. They pushed questioning. And I will say that I wrote this fluffy piece when I was looking into clinical neuropsychology programs, and this was central to my essay, which I'm slightly embarrassed about. But I wrote about how instead of looking at a sunset or into outer space or oblivion for answers about self-transcendence or of God, I only had to look within the space between two neurons and see it and feel it and measure it methodologically. And so years later now, I'm working in a brain bank. I am, I think you've heard me say this before, but I am not exaggerating when I say that I feel like I am among tiny gods lined up in a fridge. It's creepy, it's weird, I know, but it is who I am. These organs were the source of life for each and every human that made that precious donation. And for me, it's spiritual. I'm honored really daily to be among those gifts. And it's a major defining feature of not only my professional career and journey, but it's shaped me personally too.
Yeah. And a neuropsychologist, I don't think that's very common, right? These two things together. That's super interesting. Just kind of simply, what is that? If somebody was to ask you, what does a neuropsychologist do?
It's a very good question. People ask, what is a neuropsychologist? So I am a clinical neuropsychologist. And for some funny reason, neurologists, are easily understood, and psychiatrists and psychologists are understood, but the combo is less understood. And what my job is to be an expert in the relationship between the structure of the brain and the output and the behavior. What I do on a daily basis is I see patients who are complaining about something wrong with their brain, but they're not sure what. And the only way to measure the actual function or behavior of the brain is to do testing, paper and pencil, old school type tests to see how is that person's memory functioning? How is that person's attention, executive functioning, visuospatial abilities, judgment, problem solving, math, reading. We're testers. My job is slightly different because I'm also interested in the structure of the brain. And so what I like to do is combine the understanding of human behavior with the structure, with the brain structure itself. There's actually a quote that hangs in my office, and I think it sums up the beauty of clinical neuropsychology. And it is that structure without function is a corpse. Function without structure is a ghost.
And that's what you're trying to figure out within these humans. They haven't been diagnosed yet. When you see them, have they been diagnosed with dementia or Alzheimer's, or is it just that they know that there's just something that's not right?
It's both. So sometimes an individual will come into the office with a loved one, and the loved one will say, there's something wrong with my mother, with my father. I'm telling you, doctor, she's not remembering. And the patient may say, there's nothing wrong with me. The problem is him. Sometimes they come in extremely distressed, upset. There's something wrong with my language abilities. I cannot find the right words. I can't pull it out. And it's my job to investigate, really like a detective, objectively, using objective evidence and objective tools to figure out what is going wrong and why that may be.
And then when you do these tests and you diagnose, you stay with them as they're your patients for a long time. The way you've described that to me is that you're taking care of them. They're your patients, and they are with you until the end in most cases.
That's right. One of my jobs is to monitor very closely their cognitive functioning over time. So if they do come in with a diagnosis, or if they're diagnosed by me or by my colleagues, like a neurologist with Alzheimer's disease, it's my job to monitor them and to see, has there been progression of the disease? In what domains has it affected? How is that going to impact their daily life, their families, decision making, everything from finances to hospice care? Some individuals also come to me because they're interested in treatments. And the question is, if I take X amount of treatment, will this impact my cognition, Y amount? And it's very empirical.
So that's why you're more on the psychology side than the neuroscience side, because you're not diagnosing and then also prescribing treatment.
Right.
You're actually helping them through, and as part of that relationship that you have, you're working with other neurologists. You're working in concert to, just give them the best care that they possibly can on their journey.
Exactly. The neurologists rely on us for the diagnosis because without testing, you really can't know for certain whether memory is impaired versus attention versus vision. It takes a comprehensive test in order to really parse that all apart. So I would say 90% of my referrals are from neurologists that ask, questions like, is this a neurodegenerative disease based on what we call a clinical syndrome, based on the constellation of symptoms that are present.
And then circling back to the brain bank, and your description at the very beginning, and the kind of the love of and the specialness of the brain. These patients then donate the brains to science, to you, to study further their disease, and so that relationship continues even after they pass.
That's right. So at Northwestern, where I practice, and where I conduct research. We do have a brain bank. It's quite large, one of the largest in the country. And the reason why we're able to collect these brain specimens is because our patients enroll in a research project that's funded by the National Institute of Aging. Our job is yearly to collect cognitive testing, blood, imaging. We follow them from the research perspective. So I may see them initially clinically, but then they're enrolled in a research program. We follow them until they pass. And 97% of our research participants donate their brain.
Yeah, that's pretty amazing that people do that.
Amazing.
Yeah.
And it's a very high percentage.
Yeah. It's really a gift.
It is.
To all of us. So your focus on your research, your research focus, what are some of the kind of key questions that you're trying to answer, or some of the hypotheses that you're trying to test within your research, within your lab area?
So one area of interest is super aging research. And I'll talk a little bit about that. It's really very exciting. We know that in Alzheimer's disease, what happens is there's a deterioration of memory over time, and typically happens in old age, although it can also happen in younger age. And what we've began to study really in the last 20 years, 25 years, are cognitive superagers. And superagers are individuals over age 80 who function in terms of their memory performance at the level of a 50 or a 60-year-old. So when we say they're super, they really have super memory abilities. And the idea is that we're studying the preservation of memory despite old age instead of the deterioration of memory in old age. So it flips the coin of aging on its head. And it's really an incredible model to explore resilience to pathology during age. That's really how the study started by my mentors. And I've been lucky enough to work on that project. I'm most passionate about the postmortem brains of superagers. We've collected now, I believe the number 72. And that is a huge cohort of individuals who are truly superior that have given Northwestern their brain for investigation. Some questions, to go back to your initial question, some questions are basic, but they're very difficult to address. So one big question is, how are they able to withstand the effects of age? How or why are they protected from Alzheimer's disease? We've found a few years ago, we found that if you open up the hippocampus under the microscope of superagers who have passed away, you find that there is not a lot of tau tangles, which is a major hallmark of Alzheimer's disease. They really do not have that many tangles in the memory related hippocampus. So the question is why are they not forming Alzheimer's disease? Even in a typical 80-year-old, 85-year-old who is fine, who's not really having a lot of memory problems, you'll still see tangles in their hippocampus. These superagers appear to withstand that impact. So the questions, they're so vast. We can keep going. I mean, is it lifestyle? Is it shampoo? Is it the fact that they have stronger neurons? I have been asked every single question under the stars about why that may be, and it's a real mystery, and it's really powerful.
Wow. That's amazing, because, it's like, how does this, from the understanding I have from you in previous discussions, is there's really no kind of commonality as far as economics or, lifestyle, or, did they all eat a certain diet? Did they all exercise? Did they all, whatever? It's just a random sample.
Ours now is a random sample. They're rare. We're pretty strict with our criteria. And don't forget that these individuals are defined by their memory capacity. And that's a biggie because some of them don't have wonderful mobility. Some of them, may not be, working or have 18 years of education. I mean, it really runs the full gamut of life experience. And really what we're targeting here is memory. So this is different than a longevity study where we're looking at centenarians and we're looking at people who live to 100. These are people who have outstanding memory capacity. And so for that reason, I'm most interested in going for the jugular and targeting the memory centers of the brain, really looking to see what is making those structures so damn strong, so resilient. I think it's incredible.
Yeah. Is it the same if you have if you have strong memory into those age, do you still have good decision making?
Oh, good question. Good
You know,
question.
I don't know. How do these two things relate?
I would say that typically they tend to relate. If you have poor memory, you're probably not remembering step A. And so step B and C may be a little bit foggy. That's sort of how I conceptualize it. But in the brain, there's networks, they're connected, those regions. Sometimes you do see, though, individuals that have poor memory, but their decision making is really fine. That's what's so interesting about neurodegenerative diseases. Sometimes they hit one region, but not yet the other. There's a focality.
Yeah. Yeah, that's really interesting. So, and this is kind of a new area of research. How did we find these superagers, and how long ago did we even know that there were these people that were kind of resilient to getting the disease?
That's a really beautiful question. I am going to credit my mentor, Marcel Mesulam, who's a really prominent behavioral neurologist. He's one of the founding fathers of behavioral neurology. He taught me, and now I teach my students, that non standard cases teach us about standard neurobiological principles. It's such a beautiful and simple concept that the uncommon cases lead to common principles about human cognition and behavior. And so his idea was don't just push aside those few cases that deviate from the typical norm. They're actually telling you something about much larger. They can actually paint an entirely new, innovative picture. And that's what happened with super aging. So he noticed several individuals who had outstanding memory performance. It actually started with one case. I believe the case was HGR, and she passed away. He looked at her hippocampus under the microscope and he saw one single tiny lonesome tangle. And at the time, that really was a novel idea that someone who, I believe she was 92 or 94, would be able to not only show outstanding memory, but have one or two tangles when usually it's a sea or clumps of tangle pathology. So this one case got him thinking, what if there are other cases? What if there's this new aging trajectory entirely where aging can be good and not just bad?
And so, in that, how long ago was that?
That was
We're talking not too long ago.
well, probably started around in 25 years ago. That started the super aging study at Northwestern started in around 2007 formally. So we've now amassed quite a collection of brains, but also our participants. We have about 110 now at Northwestern, and they're active and they're excited to participate because I hope they realize that they have a gift.
You talked about how studying the brains of these superagers after death is both scientific but very emotional or spiritual. I think you use the word from that perspective. These personal connections. How does that influence your approach to how do you set up your experiments? How do you set up your research? How do you even, with all these questions that need to be answered, how do you kind of prioritize based on, this special connection you feel with those patients and, both living and as they've passed?
Tough question. I think first and foremost, you look for the gaps. You look for what's needed most in the research. And so super aging in general was needed in the research 25 years ago. There needed to be some kind of supranormal, not just abnormal, normal. Now, the next step to prioritize what questions to go for me, when it comes to designing an experiment, I like to set up experiments where of course there's a null hypothesis, but that by observation I have seen under the microscope with my own two eyes, I've felt it with my hands that there appears to be a difference. The research then is easy because you're setting up the experiment in an unbiased way, but with a strong hypothesis. So for example, you know, seeing that one tangle in HGR and then looking at three other cases with superior memory that then had very few tangles, when you do that in 20 cases, even if it's unbiased and completely blinded, something comes of it. So, by observation, number one, from the biological standpoint, you look for the gaps, you see what are kind of the lowest hanging fruits to understand and unravel. From the emotional perspective, sometimes it's as easy as asking a superager what they feel, what they think is their reason for becoming a superager. Many of them have said, "Oh, it runs in my family. My mother is 104, and my aunts all lives long." I mean, it's a simple answer. Others have answered things like, I ate oatmeal and yogurt every day since I was a child. And that's their opinion. And that led us to studying, a Mediterranean diet, for example. We did a pilot study because many were commenting about diet. Genetics, of course, we have to, and so on. So that's the best that I can do. Although you need students, actually. You need people to come in who are young with new ideas, who are completely unfamiliar with the cohort, and they're just asking almost like innocent questions, big questions that we're not even thinking because we're so in the trenches.
I had the opportunity to join you in your lab and in the brain bank, and I saw some of the people that work there with you, and they're fascinating. They're excited about what they're doing. They love to share their story, and it's obviously you're a great mentor because they love it there and they love what you're doing. And you just said you need those people. So, you're collaborating, you're asking them those questions. It must give you great joy to mentor too.
Oh my God. It's incredible. It's one of my favorite parts. And it's a force multiplier. It's like you inspire or you help the person find whatever they're interested in, and then they're just changing the world. And it's so fun to watch them with their creativity and their different ideas and approaches, and I learned so much from them. It's really like being a parent. I really feel that way, that same kind of joy watching them grow.
Yeah, it that was really special for me, and I appreciated the opportunity greatly. So, has there been any surprises that you've had? And I don't know how to ask the question without sounding kind of morbid, but you get to know these people, and then you get to look at their brain after they pass. Are there any surprises? I can't even comprehend what that is like.
Yes, there are surprises. And it goes back to the comment I made about the non-standard cases. So even within superagers that, let's say, are not supposed to have a lot of tangles in their hippocampus, occasionally I'll open up a brain and I'm ready to see nothing. And it is just chock full of pathology. It seems diseased, but cognitively, they're not behaving that way. Now, it's a very small sample, but when you do see that, it completely it almost like just it undoes all of your ideas. Now, there's no question that there's a correlation between a lack of disease and good pathology, but could there be some other X factor, some protective genetic factor in those patients, in those individuals, in those superagers that are almost, we call that, we call it resilience, because they have disease, but they're resilient to the effects of it. Those cases are surprising. They're fascinating. And those are the ones we oftentimes really delve deeply into because they could be holding a clue.
Yeah, that's interesting. So I've had others on that talked about stem cells and the kind of the technology that has evolved in the last couple decades, because I remember the first conversations around stem cells and using stem cells, it was pretty controversial because of how we were collecting them. And now we're to the point where we can take a skin scratch and, create an organoid or something in a dish, based on, our own cellular composition. Is there any kind of ethical questions around what you're doing or the research that needs to get done to understand this question around the disease and why some people are just not affected by the disease? I know there is more and more emphasis on ethics in research. I'm just curious as to if you've ever encountered that.
I haven't yet. That's the caveat. We're not there yet. We haven't developed the stem cells yet. I don't think we're too far away, though. And I wish I had some kind of very strong answer, but I just simply haven't been exposed to it. The consent process is so important. So ethically, from the consent perspective, especially with individuals who, I'm not talking about superagers, I'm talking about individuals who are suffering from cognitive disorders, it's a very careful, very in-depth process that requires psychologists, it requires social workers, which we are privileged to have in our center. Because of those protections, I think we haven't, thankfully, been there yet. I imagine, though, that as the stem cell field in ADRD, which is known as Alzheimer's disease and related dementia, kind of encompasses all of the work that we're talking about. I do think it will spark some, potentially it could spark controversy.
Yeah. Yeah, it's just, it's interesting. I know there's some, there's groups of people out there too that don't necessarily trust. They wonder, like, well, what are you going to do with my brain after,
Yeah.
I don't need it,
Oh,
right?
absolutely.
And those are, back to the diversity of the research. You need the diversity, you need
Yes.
those groups, and they need to feel like, well, this is what's going to happen, and it's going to be ethical and not scientific weird, right?
Yeah,
It's
yeah,
just
absolutely. And there's history of it. There just is.
Mhm.
Our social work team holds focus groups at Northwestern regularly for individuals from underrepresented groups. And the reason for that is because there is distrust surrounding brain donation, biospecimen collection, and I get it, 100%. We need to figure out what is the core, what are the core beliefs surrounding the brain donation process. And the only way to do that is simply to ask, to listen, and to compile the information to see what we can do to help educate. Because without that tissue, we're not going to have a diverse sample. It's critical. We need to do it sensitively. I mean, these are really, really can be. I shouldn't say for everyone. Can be very uncomfortable conversations to have. It's about identity. It's about faith. It's about ethnicity and race. This is about it's about death. So the conversations are very, very delicate. I do not take them lightly. I don't want to feel comfortable with it. I want to feel that I'm going into that conversation just like I'm going into the conversation with a patient who may have a diagnosis. It's serious. So I think that that empathy, that understanding surrounding brain donation is imperative, and I think we just simply have to listen to those individuals that are not comfortable and ask why, what we can do to mitigate that.
There's a stigma around, aging, around cognitive decline. I think in some cases it may be lessening, but your thoughts there, and does that impact what you're trying to do in the research you're trying to do?
Yes, 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 yes. I think that aging suffers from very poor marketing. And if you take a look at some of the pamphlets, you know, about aging and mobility or aging and really aging and diet, the front page it's like, it feels like a deterrent to wanting to age. And I spend a lot of time with individuals who are over 80 who are more engaged than I am, who have more of an active social life than me, who, I look up to in so many ways. And I kind of can't believe that that it's such a minority view. I have neighbors where my husband truly rather spend time with my neighbor who is a super ager in the study, and after bedtime he's outside hanging out with him, not me. And that's what I want. That is what I want for my family, for my friends, for my participants, for my patients. I want to be able to show the world that aging can be good. It can be graceful. It can be fun. It can be normal. I mean, it seems like aging is synonymous with abnormal.
Mm.
You know,
Mhm.
I see how people treat elderly sometimes, and I don't like it. I don't understand it fully. There is a stigma. What can I say? I
Yeah.
mean, our presidents are up there.
Mhm. I know. I
That's
know.
always been an interesting conversation too. Which we won't go there now. 2 weeks
Right.
before the election, but I mean, 80 is not old anymore. The same way it was 50, 40 years ago.
Yeah.
That's how I feel.
Well, I feel the same thing because, I find myself the age that I'm at and I sure don't feel it. It's
Yeah,
still very active and just, curious I'm sure at some point it will hit me, but maybe not, maybe not. So is there things that you would do that you wish that people knew about aging that would maybe open their eyes to it's not a bad thing? I mean, you're with these people, you see their brains, you're like, hey, here's what I wish you knew that I know.
that's a great question. There is something. So I'm gonna answer your question, not from the perspective of super aging, but actually from the perspective of trying to mitigate the risk of developing dementia. And in my field, this is something that we talk about quite frequently that I don't think the public knows about. In 2020, so only 4 years ago, the Lancet Commission on Dementia Prevention, Intervention, and Care came out with 12 modifiable risk factors for dementia. And they posited that nearly half, I think the number was 40%, 43%, I don't know, 40 something percent of all dementia cases worldwide could be prevented or delayed by addressing 12 modifiable risk factors. In July of this year, July 2024, they added two more. So now there are 14 modifiable risk factors for dementia. And I want to make sure that whoever's listening understands that when I say dementia, I'm not talking about Alzheimer's disease. Those are two different things. Alzheimer's is a type of dementia. There's many different types of dementias, including Lewy body disease, Parkinson's dementia, behavioral frontal variant dementia, primary progressive aphasia. I can keep going. Vascular dementia. So don't confuse dementia with Alzheimer's. Now, 14 altogether. Some are obvious: smoking, obesity, diabetes, history of traumatic brain injury. Some are less obvious: hearing loss, vision loss, that was added a few months ago, social isolation and depression, air pollution, LDL cholesterol. So this is really fascinating because what it means is that a common thread for all of these factors is that they are related to cardiovascular health. They're related to heart health. So what this means is that there's this focus on healthy heart is healthy brain. There's a huge literature that focuses on the relationship between cerebrovascular integrity and its impact on the emergence and spread of the plaques and tangles that occur in Alzheimer's disease. There is a relationship there. So when someone asks me, what can we learn from individuals like super agers, I say, can we pause just for a minute? I'm nervous about answering that right now because it's a pretty heterogeneous group. But what can we learn from individuals who do not have dementia? They're likely to not harbor these risk factors. I would argue that studying the exceptions are just as interesting. Those individuals that have 10 risk factors, but still are able to withstand dementia, that's amazing. So I want to start thinking about modifiable risk factors in life. They're real. And then in midlife, if you can control things like diabetic risk, obesity, smoking, aerobic exercise, you can reduce the risk of dementia.
Yeah, that's really important. It's not take care of it up on the front and then let the drugs and the therapies help for what you don't catch. But we can all do better up front to try it, because we've got this grain wave that's going to hit us. And, there's projections of just, trillions of dollars that it's going to cost with the onset of all the cognitive diseases that we're going to be faced with as a society.
Absolutely. And the push is to try and mitigate those risks in midlife before the onset of Alzheimer's disease and other dementias. That's the goal. Teach the public that in midlife to really take control over, at the very least, these 14 risk factors that can potentially really lower and reduce risk. That's something. That's not nothing. Especially from a global health perspective, from a public health perspective, from a policy perspective. I always think about it, we should be giving incentives, some kind of I'm not a politician, but some kind of incentive to our community to engage in these 14 behaviors. If it's a 40% reduction, it's gonna really translate into something big.
Yeah. Well, and individually, just not getting the disease. These
Oh
are,
my gosh, of course.
these are horrible diseases. And Do you have Alzheimer's or dementia in your family? Does it run in your family?
I do.
So you know.
Yeah, a lot of us do. It's definitely a driving force. Makes us work harder, I think, and also faster. Of course, it's for the patients, it's for for the families, and for our families, for me potentially. I hate to think about it, but I'm nearly 40, and I'm approaching midlife. There's going to be biomarkers available anytime. I mean, there already really are, but even at my age, we're going to start having biomarkers that will tell us whether I'm going to get it, and possibly when. It's going to change the landscape, and I want to get ahead of it. I want to be in the forefront.
Yeah, and I live that. I believe that too, and I try to live my life like that because it's for my son. I don't want him to see that. I don't want him to go through that with me. So if I can do something so that he doesn't have to see his mother disappear and still be there in front of him. Want to do that. So going back to, you said you do a whole bunch of tests. Not only for trying to diagnose whether there's cognitive decline or superagers, and do you also use technology? Is there? And I would imagine you do technology maybe in your lab, but what are some of the technologies that you use that are kind of on the forefront of your research?
Well, for one, we're using some really pretty incredible and sensitive digital pathology software. So, what that means is, what we used to have to do is to take a slide and look under it under a microscope, and it would take us a lot of time to analyze the architecture, and really anything that we're interested, any kind of markers of disease. Now, with automatic digitization, we're able to scan slides in. That's also incredible because we can send full slides across the world, across the globe, to have someone else analyze them in a different way. So that exchange of data is there. The resolution of imaging at the microscopic level is there. And we're working with a lot of collaborators now. I'm really excited about this, is that, you know, my feeling is that the molecular composition, so at the molecular level, the neuron, the brain cell in a superager is probably different, as I mentioned before, than, let's say, the neuron, the brain cell that's living in the hippocampus of an Alzheimer's patient. So now there are single cell genomic technologies that can reveal the actual cellular composition, the identity, the states that the tissues are in, whether it's damage, whether it's integrity, how strong they are. I mean, you can even visualize shape and structure at really incredible resolution. And so we can do the same also at the protein level. And as we know, when I talk about pathology, plaques and tangles, they're really just a bunch of proteins that have kind of twisted and gone awry, and killed the cell. So if we can understand at the protein level, at the molecular level, at the genetic level, what is happening at the single cell level in these tissues, we could develop a tool potentially to ensure protection of that cell that would otherwise be targeted by disease.
That and that will speed up the kind of the discovery and the understanding, and possibly these tools or therapeutics.
Oh, majorly, majorly. In fact, only a few years ago, we had to actually look at just a single cell, or we had to take tissue and mush it up, and then look at a bunch of cells. But now, this is amazing. You can actually look at the cellular composition, or the genetic composition as it sits on the slide itself, and just project that onto a screen and kind of click and say, I wonder what molecular profile does that one single neuron have in the hippocampus? Well, 2 millimeters over in the area that is not affected, what is that neuron expressing? I mean, my God. So the technology has completely just accelerated the research and the opportunities with these brain tissues to an incredible level. And actually, it's kind of hard to keep up with the technology. And you must have collaborators that are complete experts in that field. This is the way to accelerate it, because I have my own expertise. They might not even know the difference between memory and attention, or a superager, or the hippocampus, or some other region, but man, do they know the technology. So when you bridge the two together, that's really how you push things forward.
Yeah, I was going to ask you, how do you even keep up? And,
You can't be an expert. I really do believe this. You can't be an expert, a true expert in 10 different disciplines. That's how I feel. I can be familiar. I can be comfortable. I can kind of talk some of the language, but to do the real work, you need to pair up with someone and kind of have the humility to say, that's not my jam. That's okay. I can contribute something else. It's why team science is so fulfilling when you find the right people.
do you see that there's going to be more of what you call team science because of this acceleration? Is it going to change how we do research? Because I know researchers in the past, they kind of like to do their thing. They like to do their thing.
The problem, actually.
Yeah. So say more about that. What do you see for maybe the next 10 years around how will research change, or the process of researching change?
That's a big question.
With no right answer.
Yeah, no right answer. And after I just said that, I'm not an expert in that domain or that domain. But here's what I'll say about Alzheimer's in particular. Let's stick with a paradigm. So there are now treatments for Alzheimer's disease. They target the amyloid protein, and I don't want to get into specifics here. They were controversial. It's a whole conversation, but they exist. What is promising to me is that the technology to modify a target, a disease target in the living brain, is available, and it works. The patients and the cohorts are ready to be enrolled if and when a drug comes out. They're there. I'm meeting them daily. There's momentum, there's intelligence. In the next 10 years, my hope is that there will be, like you said, an integration of expertise. For example, neuropsychologists who understand function, who understand the living experience, genetics, proteomics, pathology, anatomy. And I think that if you're able to bridge those techniques to understand what's happening at the cell level, where the disease is actually happening, where I can see it happening, I think that there can then be a personalized approach to Alzheimer's in stages. So, for example, depending on your genetic phenotype, your background, on your history, can we first increase your immune response, lower your amyloid, strengthen the cell walls, prevent damage to the cell with a specialized enzyme, and then target tau. So it's as if we are going to follow the progression as we know it exists from cell to cell and tailor it to the individual. My hope is that enough experts are going to come together, figure out how to speak the same language to fulfill that goal that helps the individual.
That would be amazing.
Amazing, amazing. And most importantly, it has to happen fast. It has to happen quicker.
Quicker. I mean, it is. I think sometimes maybe the institutions get in the ways, the way we fund research gets in the way. I don't know your opinion of that because you're right in the middle of it, but it's the old way of doing business that has momentum that has to be challenged.
I agree. I think we can learn a lot actually from the corporate space. I think we can learn a lot from different kinds of other communities that are moving really fast, grassroots organizations. It may be time for an overhaul, for a renaissance, shall I say, just to really bring people together and move faster. I really feel that way.
I like the term team science. I like that.
Yeah, absolutely.
I really like that. So as we kind of wrap up here, this has been great. I am fascinated with the superagers. I see people that are physically strong and they're bodybuilding at 80 years old. They're deciding to pick up, how to play the piano at 105, whatever these people are. But the cognitive piece and the cognitive health is so important. And it's just fascinating. I think, like you said, we have a lot to learn from them. But as we kind of wrap up here, are there things that you wished I would have asked so that you get that message out?
I think what's very important in research is not to ignore the emotional component. I think it's okay to be a nerdy scientist with emotions, who cry in a lab, who feel for their specimens. I think a lot of times there's a perspective that science needs to be devoid of feeling. I don't know where it necessarily came from. Maybe because it's that analytic, that logical side of the brain. But what I've found is that the individuals that I work with who mourn the loss of their research participants, who get off the phone and need to kind of console each other after a difficult conversation of brain donation. Is it more difficult? Yes. It makes the work, I think, more difficult, more emotional, if you will, more attached. Maybe that's the better term. But I think it's needed.
Yeah, I appreciate that. I just really appreciate that. Like I said, you gave me the opportunity to go and hold a brain in my hands, and I was
Yeah.
surprised.
Yeah.
I was very surprised at how I felt. And we shouldn't be afraid of it, and we shouldn't hide it either. We shouldn't,
Exactly.
we should
Exactly.
celebrate it. It's a part of, it is a part of life, and death is a part of life. So I think we hide that fact. We try to, hide it. So I do really appreciate those words. So Tamar, how do people get a hold of you, to learn more about what you do, to maybe, volunteer to be tested as a superager? Like, how do they, how do they get a hold of you?
They are welcome to find me online at brain.northwestern.edu. That's the website for the Mesulam Center for Cognitive Neurology and Alzheimer's Disease. And feel free to get in touch with me, Tamar.Geffen at northwestern.edu.
That's great. Thank you so much for your time. I know you've got double jobs, right? Being a clinician and working in the lab. So your time is very precious, and you've just you've shared this with us today. So I appreciate it. Thank you very much.
Thank you so much for everything that you do. Really.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes, and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.